My name is Romain Fontain, so I'm a researcher at the veterinary school. I'm sorry, my talk will be in English because if I try Norwegian, you will all leave in the next seconds. So I will try to convince you that small model animals can be used to investigate research questions linked to aquaculture. So in my opinion, it is important to understand, to have a good knowledge of the fish biology if you want to improve the welfare or the techniques or the protocols to raise fish for aquaculture. And fish, of course, are not all the same. They are different depending on where they come from and the lifestyle that they have. But they're also similar in many aspects. And if you think about this fish here, that shares about 70% of its genetic code with humans, you can easily understand how fish here are similar to each other. And in some cases, it can be interesting to use another fish species than the one that you are interested in, if it provides faster, cheaper access to the answer that you are looking for. And for example, this model fish here, Medaka, which is highly related to salmon, has a much smaller genome, less than a third, less complex as well. That makes, for example, biology studies much easier in this species. So when I'm talking about small model fish, I'm mainly talking about zebrafish and Medaka. These two species, they have a short life cycle, so they live, they are adult in less than 3 months. And they are small. They are 3-4 centimeters long, so you don't use a lot of food, and you can put many fish in small tanks. So that makes quite relatively cheap to raise and maintain these fish in the lab compared to bigger fish. And this is, for example, a picture from the fish facility that we have in the new veterinary building. So you can see that we can raise a lot of those fish. And we have specific, if you don't see my specific equipment where we can control several factors of the environment, such as light, temperature, salinity. So we can study each factor separately, but also in combination. And that provides quite interesting clues about the way the biology works in the fish in a really controlled environment, which is difficult to get for bigger fish species. And that's just to finish an example from our lab. So at some point, when the fish grows, they will go under sexual maturation. And so they will use some of the energy to grow the gonads instead of growing themselves. And this gonadal development is controlled in all the fish the same way by what we call the brain pituitary axis, where the pituitary is mainly secreting these two hormones, FSH and LH, that stimulate gametogenesis. And this is FSH and LH over time in the salmon. And you can see that FSH comes first, and at a later time points comes LH to stimulate the last step of gonadal development. And if you look at Medaka, the light blue lines, you see exactly the same thing. First come FSH, and at a later time points comes LH. The only difference between these two species is that the time scales. Here we are talking about several months, almost a year. Here we are talking about 14 days. So basically you can answer the exact same mechanism in a much shorter time. So that means that you save time and you save money. In addition to that, you have a lot of tools because the research community working with these animals is quite huge across the world. And there are many new tools developed all the time. And this is one of the tools that we have developed in our laboratory, where we have developed transgenic lines where these two cells that I was talking before that produce these LH and FSH cells, LH and FSH hormones, we have labeled them with a fluorescent protein, FSH in one color, LH cells in the other color. And we can investigate these cells either in vivo, in vitro or in vivo in the fish itself. And we can, thanks to this labeling, we can visualize the cells, localize them, and so we can identify their role, their function, their regulation, something that is impossible to do or really complicated to do in a bigger fish such as salmon. And one of the last findings that we had in our lab is that we identify that the number of these cells is strongly regulated by the photoperiod, so the length of the day, and by the steroid hormone that you find in the blood. So why is that important? Because the more cells you have here, more hormones you will produce, so more you will induce gonadal developments, and then less you will have growth. So understanding what regulates and controls the number of these cells and their activity will allow you to understand, to have a better understanding of the gonadal development and so the growth. So just to quickly conclude, small model fish, they can be very good tools because they provide quick answers and for a cheaper price. And thanks to the number of tools that exist, you can usually have a deeper understanding of the biology of the animals. But they cannot replace the research in the aquaculture species. It's not my point. I don't say that they will save the aquaculture world just by using model fish. What I say is that they are complementary, and often they can be used as tool to go faster, to go cheaper, and to target then your research in the model animal, in the animal of aquaculture. Just a last slide, just to show you that in many pharmaceutical companies, they have started to understand some years ago the benefit of using model fish, even to study human health. So many of those labs, pharmaceutical companies in USA and Europe, they have started to develop their own fish facility, working with zebrafish mostly, where they try drugs on these fish because it's cheaper and it's faster to see the results before to go even in mouse. And yeah, most of them have integrated and developed such labs in their own facilities. So in my opinion, if these companies have seen the benefits of such model fish for their study on human health, I think it should be much more relevant for aquaculture. Thank you very much.